doesn't occur, the worst encounter is that it's actually going to interfere with the signal that's coming for the next go-round. And again, these are cumulative processes, so they may happen a lot over the course of the sequencing reaction. They produce noise, and that can cause errors and ultimately limits the read length as well. So just always be skeptical about how well each of these steps work, because there are places where they fall apart. Okay, Illumina has been, I think, in my opinion, a pretty remarkable company also just in terms of the amount of data produced from these instruments. So I don't have a comprehensive listing here of all of the, the early selected basically produced about 1 billion base pairs per read, of single-ended reads. The newer iteration, what Illumina took over, was the GA2X. We then, in early 2010, encountered the HiSeq 2000, which ran 2 flow cells coincident with one another and produced on the order of about 200 gigabases per run in about an 8-day period. And then the most recent version of the HiSeq, which was announced, or released rather in July of this past year, is the equivalent of 6 whole human genome sequences per run of about 10 to 11 days, as I mentioned. So this is a remarkable jump up in terms of the quantity and actually also the quality of the data that results from this. And in our laboratory, this is the primary sequencing instrument that we're using. There's also a newer version called the MySeq, which I'll talk about in a little bit, which is a personal genome sequencer, if you will. It's sort of a desktop instrument, much lower scale, as you can see from the numbers here, but I'll get to that in just a moment. And then the last thing to mention on the Illumina technology is, as I mentioned, there were some recent announcements in January about improvements to the different sequencing platforms. Illumina's announcement was for the 2500 sequencing instrument, which is basically not just a modification of the HiSeq instrument. In fact, you would be able to upgrade the instrument itself, and that will produce less data, about 120 gigabase pairs per run, but the run requires only 25 hours to complete. So now you're talking about the rough equivalent of covering a human genome in a 1-day period to generate the data. And we don't have those instruments yet, but we're beginning to look at data from them. I should point out that nobody has them yet, so they're somewhat vaporware at this particular point in time. We shall see. Error rate is, yes, thank you. I forgot to mention that. The error rate has been improving pretty dramatically over time. Originally, we were around about 1% error rate on this platform when we first started working with it. The recent version 3 chemistry that I mentioned is down around 0.3% error on the reads, and we also are seeing a much better coverage on the G plus C content, where in the past, very high G plus C sequences of 95% higher actually did not represent well in the Illumina data set, and that's now been addressed by some changes to the chemistry that we saw with the version 3 release. So that's improved the coverage overall in the genome as well, which has been a relief because it was pretty easy to see. We published a paper in 2008 that actually showed that you couldn't see these sequences. Okay. So the third large sequencing technology for human sequencing and other approaches is from a company, Life Technologies. This is a different beast, sequencing by ligation, and what we've been talking about earlier, which is sequencing by polymerase. We use a custom adapter library, as I mentioned. This is also an emulsion PCR-based sequencing instrument, and Life Technologies actually has some nice modular equipment that you can buy to facilitate the emulsion PCR steps, because they are pretty manual if you don't have that instrumentation and subject to some errors and failure points, and those instruments need to help. So this is sequencing by ligation. The bottom line is that we have fluorescent probes. They're about 9 bases in length, and they have a very defined content, and we are also priming from a common primer once we have these emulsion PCR beads to sequence from. So rather than sequential rounds of base incorporation, this is now sequential rounds of ligation of the primer. A detection step takes place. There's some enzymatic trimming of the primer that was, or of the INRO that was added on, and then a second round of ligation follows, et cetera, et cetera. So we go through sequential rounds of ligation. We also go through sequential rounds of primer. So when your first sequencing primer sits down on the adapter at the N-0 position, the second, and you sequence bases 5, 10, 15, 20, et cetera. The second go round, your primer sits down at N-1, and you sequence bases 4, 9, et cetera. So it's sequential and sequential, if you will. The beauty of this approach is that in the design of the ligated 9mers, you effectively have the first 2 bases identified with a specific known sequence, and those correspond to the fluorescent group that's on that particular 9mer. So why is that important? Well, what it means is that you're effectively sequencing every base 2 times, because these first 2 nucleotides are fixed, and their sequence is known from the fluorescent group that's there. So effectively, if you can look at this particular little diagram here, if you can't see these well on your slides or on my slides, for any of these approaches, I would suggest very strongly going to the manufacturer's websites, because they have extraordinary, sometimes animated visual aids to help you understand the unique attributes of their sequencing process. But effectively, by sequencing 2 bases per cycle on each incorporation, you end up overlapping the bases that are read from the fragment. And so you effectively sample every base twice. I like to refer to this for a common analogy is when you write a check, you write in the number of the dollars and cents that you want to pay on the check, and you write it out secondarily in longhand. So there's that ability to sort of cross-compare, if you will, from one read to the next read to make sure you've gotten it right. And this yields an extraordinarily high, or extraordinarily low, rather, substitution error rate for this technology. I'm going to have to change that analogy because not many people write checks anymore, but so far it's still working, I guess. So this is a solid instrument is the name of the platform. These are the two most recent versions, with the 5500 XL being most recently introduced just last year. And these are some of the attributes. As you can see here, the error rate is extraordinarily low. We have this front-end automation, a 6-lane flow chip that actually allows you to use some lanes and not other lanes, sort of to ameliorate this need to sort of load everything up at once if you don't have enough samples, etc. And the very high accuracy, as I mentioned. And they're introducing some new primer chemistries. I'm not sure that those are actually out yet, that would increase the accuracy pretty wildly high. So that could be, I think, very interesting. Okay. So let's shift gears now away from next generation massively parallel instruments to what are commonly referred to by some as third generation sequencers. And this is really more than anything to just denote the time point, which was sort of the beginning of last year when these sequencers started to hit either early access or to actually hit the marketplace. So these include the Pacific Biosciences sequencer, which is the first true single molecule sequencing instrument that I'm going to talk about. So we'll see what the specific attributes are for that system, which basically marries nanotechnology with molecular biology. The ion torrent system now is a variation of pyrosequencing that instead of detecting light, actually detects changes in pH, because not only is the pyrophosphate released when you incorporate a base, but a hydrogen ion is too, so you effectively can monitor base-by-base changes in terms of whether an incorporation has happened by monitoring the pH in effectively a little modified semiconductor apparatus. And then the MiSeq I've already mentioned, which is really just a scaled-down version of the HiSeq with very great similarities in terms of the process and the chemistry, etc. These all offer some shared attributes as well: faster run times, as we'll talk about, lower cost per run, and a reduced amount of data generated relative to the second-gen or next-gen platforms that I talked about. And also, some are touting a potential to address genetic questions in a clinical setting because of the low cost and speed with which results can be returned from these instruments. And I probably won't talk about that, but during Q&A we can address that if you're interested. So I put these in with the other systems just to place them in context along the lines of what I just talked about, different detection methodologies, how the libraries are made, and you can see here from the Pacific Biosciences that we don't really require any of these concerted amplification steps, although in the true sense of full disclosure, there are some PCR steps involved here as you make the libraries with the specific adapters. And then the run times, as you can see, are quite low, 45 minutes, 2 hours, and on the order of about 19 hours now for the MiSeq platform. So let's go through these step by step. I just put the top bio here first because that was the first third-gen instrument that we received in our laboratory. And you can see, again, just sort of very common shared steps here relative to the next-gen platform. So the sample prep requires shearing, polishing at the end, and ligation of a specific adapter that's called a smart bell. I'll show you in the next slide exactly what that is. Smart bell is obviously just a marketing name for it, but it's kind of a clever little adaptation. And then sequencing, primer annealing takes place where you actually bind the DNA polymerase to the library molecules first, and then introduce them onto the surface of the smart cell, which is a little nano device, essentially, that contains on the order of about 150,000 so-called zero-mode waveguides. I'll tell you what those are in just a moment, but you effectively